Okay, guys, some more notes from that I took during my faculty meeting today. I know they're messy and jumbled and disastrous to look at, but here I am. Hello. So, let me just bring your attention to some things. Next up, cancer. So, cancer, awful, terrible, but important to understand because most of us are affected by it or know someone who is. So, genetically encoded, oftentimes, right? We're talking about cells that are just undergoing uncontrolled mitosis. That can be disastrous because of a number of characteristics, right? They lack contact inhibition, they bump into stuff, they don't care, they start doing damage. They oftentimes will revert to immature forms, and we know that mature cells need to be mature to perform certain functions. So, a lot of times cancer results from genes that are essentially responsible for controlling the cell cycle and for creating specific proteins that travel around your body performing functions that eliminate cancer, right? Check it for mutations, eliminating cancerous tissue, and so forth. So, genetically encoded, so this crazy word right here says. Remember the word proto-oncogenes, right? Proto-oncogenes are the good version. Oncogenes are bad, right? Proto-oncogenes create proteins to regulate the cell cycle properly, and if they mutate into oncogenes, then essentially they're no longer doing their job. We can end up with cancer in that particular region, right? Remember, we discussed all those oncogenic factors. A lot of viruses and so forth are responsible for causing proto-oncogenes to mutate into oncogenes and thus can cause cancer in and of itself, right? So these things make growth factors. These genes are responsible for creating proteins to regulate the cell cycle and certain growth factors as well, right? Remember, we have growth factor genes that are responsible for producing enough growth factors to essentially control the cell cycle that we discussed. Remember that gap stage 1 and gap stage 2 and the S phase, and all of those are part of the cell cycle. And without going into too much detail, if you ever take the higher level biologies, we go into all of the details of exactly how the cell cycle is regulated and certain concentrations of growth factors and so forth are responsible for this. Just know that cancer occurs as a result, oftentimes, of mutations in genes relating to the cell cycle, right? That's all you have to really take away for that. So, cancer is immortal, lives forever, right? We talked about the HeLa cells, which are just those cells that, let's see if I can move this crazy. Not that it really matters, since you just really need to listen to my voice. But in any case, lives forever. So that says right there. Normal cells die after about 50 divisions. That's what apoptosis is. And most cancerous cells, if the body's taking care of what it's supposed to properly, then those cancerous cells will become apoptotic, and the cell will eliminate any cancerous growth on its own. So, we talked about the HeLa cells that were established as one of the first and still established cancer cell lines from Henrietta Lacks. Why are cancer cells immortal? Well, that's because they rebuild their telomeres. So this says right here, right? They have that active telomerase enzyme where they can continuously build those protective caps on the end of chromosomes to shorten after each cell division, which is why cells actually commit to apoptosis. It's when we start getting into those coding regions of the genes on those chromosomes, but cancerous cells can rebuild those protective caps, those telomeres, with that telomerase, active telomerase enzyme, and therefore they remain immortal, right? Other key factors about cancer: they thrive in anaerobic environments, right? This is a really key, strong point to this. Remember, we discussed PET scans, these positron emission tomography scans, and these are essentially just scans where we inject this radioactive dye into an individual, which is just a radioactive isotope that's piggybacked on a glucose molecule. We know that we see glucose entering in cellular respiration at the stage of glycolysis, right? And in fact, what's really interesting about cancer cells, they also secrete specialized enzymes that enhance glycolysis, because as you are cancerous tissue and you're sucking up all the nutrients and oxygen in an area in which you're growing, right? Maybe you have a tumor that's starting to rapidly grow. The growth rate is so exponential that essentially that area is starved of oxygen, and hence having specialized enzymes that adapt your ability or adapt the cancer's ability to perform glycolysis at a higher rate allows that cancerous tissue to thrive and grow much quicker, right? And we use those PET scans to look for areas where glycolysis is occurring the quickest, to see where that radioactive glucose is being taken in that anaerobic environment, and therefore we're seeing where localized cancer tissue may be occurring. So that's one of the most invasive forms of detection. First, obviously, we do preemptive tests to try and see if we need to do something like a PET scan, right? We talked about pap smears, we talked about mammograms, we talked about colonoscopies. You guys are familiar with all of these, right? And the specific cancer types that they are used for detecting. If you're not, make sure you go back and review your lecture guides. You should always refer to your lecture guides if you're not sure what the heck I'm talking about. Right? So you have these early detection methods. If you're concerned, if your doctor is concerned, usually they'll refer you to an oncologist, and then and only then would you be referred to have some type of PET scan, right? So, the treatment of cancer. Most of us are familiar with these treatment methods as well, right? So for localized cancer, remember, stages, grades, and all those things. I don't need you to know any details about those whatsoever. Just know the stage is essentially assessing the spread of a particular cancerous region, and the grade is essentially just how recognizable that particular tissue is compared to healthy tissue from that area, right? That's all you need to know. Nothing details about grades or so forth. Just more or less important information for going through life, right? So treatment. We're familiar with radiation. If we have small localized area, then you can surgically remove it. It says surgery right here. And you can treat it with radiation as well. And of course, a lot of times radiation will be followed up depending on the cancer type and the probability of that particular cancer spreading. Fancy word for spread is metastasizing, right? Then you'll have chemotherapy treatments, which is essentially just a suite of a number of different drugs, oftentimes injected and taken orally, that essentially target cells throughout your body that divide quickly, right? And that's why if you are undergoing chemotherapy treatment, all cells that divide quickly are targeted, right? Remember how chemotherapy drugs work. So most of them, you see this sentence right here, interfere with spindle fiber formation, work by essentially interfering with the spindle fibers, which we know essentially structurally, or they're cellular structures that maintain the integrity of the cell. They're also responsible for moving organelles around and for moving chromosomes around during cell division. So they interfere with the spindle fiber formations. Now, there are some newer methods. We also mentioned a couple other ones, right? So if you can't have localized surgery and then you follow that up with radiation treatments, right, just bombarding these cancerous cells with free radicals in an effort to cause them to die off, then of course you have chemotherapy just to treat metastasized cancer and to eliminate all cells that die quickly. Obviously, that's the problem with the race of cancer treatment, right? You're trying to kill off more cancerous cells than you are quickly dividing healthy cells, and that's the battle. We also talked about some more modern methods like the inhibition of angiogenesis. Remember, angiogenesis is just this really fancy word that means blood vessel growth and development, and if you inhibit the blood vessel growth to a particular region where we have something like perhaps tumor growth, for example, then you can essentially starve the nutrients that tumor need to survive and thrive, right? So we do this in some treatments, not in others. We talked about the benign fibroid tumors quite a bit this semester that females get, and nearly half the population of females in the U.S. suffer from fibroid tumors at some point. Some are too small to detect, some are large enough to need treatment. Angiogenesis is one way they use, or the inhibition of angiogenesis is one of the ways they use to treat that, right? So a lot of times they can actually ligate or cut off the blood supply in the uterine artery. So anyways, sometimes we just try and stop the blood flow to a particular cancerous region. We talked about more modern treatments, right? Some of the more promising treatment methods for cancer now and in the future include cancer vaccines, because the only thing essentially that can recognize cancer in our bodies is our own immune system. And we kind of ambiguously group these treatments into both direct and indirect methods of cancer vaccine treatment, where essentially indirect treatments are using those cytokines, or essentially those secondary messengers that our immune system uses to label things, right, and essentially teach our immune system what to recognize. We looked at that animation of using that virus to essentially insert genes that you would commonly find on the surface of certain types of cancer to teach our immune system to recognize those cell surface proteins so that they know specifically what cells to attack. And then we talk about direct methods of using cancer vaccines. We talked about some of these things where we inject this compound called Rexin G, which is just this gold-plated nanoparticle essentially coated in the opposite sequence of oncogenes, where essentially this gold nanoparticle is circulating throughout the body. It permeates cancerous tissue growth, and then it causes the destruction of that cancerous tissue growth because gold nanoparticles are in high enough concentrations toxic, right? Then we talked about these two Tykerb and Herceptin treatments that essentially target cells that have higher concentrations of that HER2 receptor that we typically find on all cells, but some types of cancer, breast cancer for example, those cell types have a higher concentration of HER2 receptors. So with Tykerb and Herceptin, we just have this cytotoxic compound that's linked to this antigen that binds with the HER2 receptor, and then that's imported into those cancerous cells, and that cytotoxic compound poisons those cancerous cells, breaks down the DNA, for example. And in that way is another good example of perhaps what we can call a direct cancer vaccine treatment method, right? So we also have prophylactic vaccines we've discussed before, like Gardasil and Cervarix and so forth. But all in all, that's just a good breakdown of essentially we have cancer, how it's regulated, the genetic level, some attributes of it that make it so virulent and dangerous, some attributes that make it live forever, how it can thrive in both anaerobic and aerobic environments, though particularly it's specialized for anaerobic environments because it tends to create them on its own, the ways that we originally detect cancer, and then, of course, how we detect its possible rate of spread, and then some treatments that we can typically use in a hierarchical method all the way up through chemotherapy, and, of course, more promising modern treatments for cancer.